All right. Today I'm going to go over this 1990 paper by Zametkin et al. It is the first paper to definitively, well, to provide very strong evidence for the existence of ADHD. And the very brief summary is that they measured glucose uptake levels in the brain throughout different regions using PET scans and found that in ADHD patients, the glucose uptake, the relative glucose uptake was significantly lower in the frontal regions relative to controls. And this is important because these frontal regions are responsible for, planned complex motor movement, social types of behavior like impulse control, things like that that are associated with ADHD. So let's get right to it. I'm going to share my screen. And yeah, so we should be up. Okay, so here is this famous paper published in the New England Journal of Medicine. And I'm just going to go through section by section and read bits and just summarize things. So the abstract is basically what I mentioned. They include much more details on the statistical analysis with the p-values. But let's see. So I'll get into some of these more as we go along. All right. So first, the background. In all of these papers, if you haven't read papers like this before, scientific papers, the introduction sets the background and it leads up to a gap statement. So they summarize, okay, this is, here's the really broad picture, that's the start of the funnel, and then they start talking about the studies that people have done within this broader context, and they start to narrow down into the question that they're looking at. So they knew at the time in 1990 that hyperactivity is a disorder of unknown cause that affects 2 to 4% of school-age children. And people didn't quite yet understand there wasn't quite any strong evidence for brain differences between hyperactive children and regular children. So at the time, the validity of hyperactivity was controversial. And in the study, they say we used PET scans. And I'll get to some more of the details of that in a bit. And they chose adults because PET scans include ionizing radiation. It's not, it's not incredibly dangerous. It's more than an X-ray, but they just to be absolutely safe, they said we're going to choose adults for this study. And children are also they're a little more, they're just naturally more sensitive to radiation. So it's just a decision that made sense. All right. Symptoms of hyperactivity are motor restlessness, attentional difficulties, distractibility, and impulsiveness. So the disorder is probably inherited in certain families, as reported by Biederman et al. And so they knew at this time that it seemed to be genetic, even though the validity of hyperactivity was controversial. And symptoms persist into adulthood in 40 to 60% of persons. So I believe this number is higher. In other words, hyperactivity in adults is still underreported, and it's a much smaller percentage of hyperactive children that actually grow out of it. All right, so eventually we'll look at journals that publish studies that established studies that confirm that. And right, no consistent finding has emerged to implicate a specific neurotransmitter, modulator, or neuroanatomical substrate. So people try to hunt down specific neurotransmitters and specific mechanisms that could cause hyperactivity, and they were not successful at this time. Earlier neuroanatomical imaging studies of patients with attention deficit disorder with hyperactivity either have found no abnormality or have included large numbers of subjects with substance use disorder. So major confounds in imaging studies. And so also they're starting to narrow down that funnel, right? So they started with broad definitions of hyperactivity, some things that are known about genetics, and now they're turning to the imaging studies, which is what part of this study is about. Now, right, so there was a previous study that measured cerebral blood flow abnormalities in ADHD patients versus controls. But again, there were some confounds in this case. large age discrepancy. And the study included subjects with developmental dysphagia and mental retardation. So if I recall correctly, dysphagia has to do with reading. Some people, either language or reading, people have trouble learning and understanding language. So, some kind of learning disorder. People with learning disorders were included in the study. Major confound. To improve on these earlier studies, we examined cerebral glucose mechanism rather than blood flow and used a scanner with considerably higher resolution. So right off the bat, they propose major improvements to prior studies just by the imaging. The way that they're imaging and measuring the amount of brain activity is already very different. And then they also, in the methods in the sample, we'll go into this in more detail, but they also mention that they're careful to eliminate as many confounds as possible. In addition, the patients in our study had not been treated with stimulants and had no history of substance abuse. We decided to study hyperactive adults who were the biologic parents of children given a diagnosis of attention deficit disorder with hyperactivity. So I don't think the familial hyperactivity, increasing the likelihood of identifying a biologic determinant, I don't think that was part of the study, but I think they included it just in case future studies need it. That sentence is a little hard to parse, but it's part of this. Yeah, so I think that's it sounds like it's future proofing. And just say something if I'm mistaken. As children, these parents were themselves hyperactive. Yeah. So the controls, the ADHD patients chosen for the study are parents that have hyperactive children, and then the parents themselves as children were also diagnosed as hyperactive. And they also explain the criteria used as the Utah criteria or criterion, which was for a long time was a standard set of criteria used to diagnose hyperactivity. All right, and another mention of genetic links. And the parents in our study continue to have symptoms as adults, and with 3 exceptions, were living with their hyperactive children. All right, so very good. You really do try to, at least even in this brief summary, you can get a sense that they are trying to homogenize the samples that they have. All right, so more details. 50 healthy normal adults, 28 men, 22 women, participated in the study as controls and were assessed in a semi-structured psychiatric interview. And yeah, they go into these details. They measure, they take note of a ton of different things, right and left-handedness, the past severe medical or psychiatric problems. I believe they excluded, so they excluded patients that had serious psychiatric problems in the past. 25 patients were included if they reported a definite childhood history of attention deficit disorder with hyperactivity. And yeah, they used the Diagnostic and Statistical Manual of Mental Disorders, or the Utah criteria. Yeah, it met the Utah criteria. Let's see. Then they take note of a ton of different things. So in this table, they note down that they note the age, sex, estimated IQ. They take these reading, spelling, arithmetic tests. They rate their parents, self and spouse on the degree of ADHD, and things like education level, alcohol consumption, police record. So they want to eliminate people that have a strong criminal history, because then you might just be measuring brain differences in criminals, people that tend to be more criminals than people that are not. Alcohol consumption, they don't want to measure ADHD levels in people that are alcoholics versus not. So very helpful table, and this is an example of what makes a good study. They're very transparent about the samples that they have. At least there's a strong attempt to be transparent. That's very important. Okay, socioeconomic data. All right, so that's the sample. Good homogeneous sample. The only criticism that I have, by the way, as someone who's naive, I mean, my training is in applied mathematics. I'm not trained in designing and carrying out experiments. I did spend a good amount of time in a neuroscience lab, worked alongside experimentalists for a good 10 years, and observed a lot of experiments, but I wasn't the person to actually execute and be in the trenches and understand the subtleties of experiments. So I'm probably missing something more subtle in the samples here. Keep that in mind. And if you do have experience in experiments, I hope you'll say something, comment if I miss something important. So as far as I can tell from my naive perspective, the choice of samples, the way that they filter out they select the samples is very strong. On the other hand, the sample size is relatively small. And I don't know if this is an acceptable size for human studies. It very well may be. I don't know. All right, and then next they use positron emission tomography. Very, very strong, robust tool, I would say. It's relatively low resolution compared to more modern things. The CT scan is higher resolution, and I believe the CT scan was only starting to come around around this time. But I don't know if CT scans, so I don't know about the details of CT scans, but I do understand the details of the PET scans. And for the time, at least, it's a very good, relatively high resolution method. So the way it works is patients are injected with radioactive glucose. Again, the radioactive dose is very small. It's not like, they're not going to turn into Hulks because they get injected. They're not going to get cancer. It's within the level of cancer that they could get it's so low, the percentage is so low that there's a lot of statistical noise, and it's very hard to tell if PET scans could even cause cancer. There are ways to estimate it, which I can get into later, but anyways, they're injected with radioactive glucose. The body recognizes this glucose as any other, just like regular glucose. The radioactive glucose looks the same, but it has one, I think it was a hydroxyl group. It's replaced this oxygen-hydrogen branch that comes out of the molecule that's replaced with fluoride. And the fluoride will decay. It'll release, I believe it was positrons. Yeah, it releases positrons. Okay, so even before I get to that point, the glucose is absorbed by parts of the body that have high metabolism, that really need a lot of sugar. So cancers, they require a lot of energy. I believe something like the heart, maybe the kidneys, some organs have very strong glucose uptake, and then the brain, absolutely the brain. So the brain will take up glucose in particular regions that need it the most. And if that radioactive sugar is taken up, and as the radioactive sugar decays and emits these positrons, you can use that to locate precisely where the glucose uptake is highest. So as the sugar starts getting sequestered, they emit these positrons. They're emitted very quickly at first, but they lose momentum. And within a very short distance, this happens on a tiny scale, they hit an electron and they get destroyed, and they emit these gamma radiation, these gamma photons. And once they hit and they're eliminated, they emit gamma photons in almost 180 degree on almost perfectly opposite directions. So if you surround a human or whatever with an object that can detect this type of radiation that's getting emitted at 180 degrees, you can actually get a good idea of the line from which that activity came from. And then using some statistics, which I don't quite understand, I'm happy to learn more about that if there's interest, they can actually estimate the actual position along that line where the event occurred. So really cool, there's a lot of really cool physics involved in that. And that's the basic idea behind it. There's radioactive decay, and it's detected by some machine, and you can pinpoint the approximate location of where that sugar was, where a relatively large amount of sugar is being used, which correlates to brain activity. All right. Raw pixel values. So they're also very detailed about how they convert the raw data to the actual plots. And the next part is they have an auditory attention task. So the level of glucose uptake in the brain in different regions will depend on the type of task being done. In this case, they are doing an attention task, and the glucose will be taken up most in brain regions that are being used most. Makes sense, right? In the task, the subjects are blindfolded, and they are instructed to listen to 3 tones of varying frequency. And for example, if as you're blindfolded, you might hear do, do, do. Then you indicate which of the 3 tones was lowest. In that example, you would indicate that the first tone was the lowest in frequency. So very straightforward attention task, and all the subjects were trained in this task. They knew what was coming. Everything was done so again that they're trying to homogenize the samples. Okay. And interestingly, let's see. Okay, so I think so that's just the data method statistical analysis. So I should also say that my statistical analysis isn't the strongest. There are lots of things that I recognize at a surface level. I've done different tests for classes, but I'm not trained. I don't do statistics for research. So that's another thing. If you are good at that stuff, you should. And if I miss something important, please say something. All right. So the results. Here's how they present the results. Left brain, right brain. The PET scan takes information from not just the entire brain, but you can actually pinpoint relatively accurately what the activity levels are in different regions of the brain. So to get a better idea of which brain regions are more active or less active, they chose bins, and they just said, okay, this brain region is, this bin goes here, this bin goes here, and they measured the glucose uptake levels at each of those bins. Left brain, right brain, and yeah, this just explains the different regions in the brain. Okay. Results: glucose metabolism. Assessed by PET scanning, global cerebral glucose metabolism was 8.1% lower in the hyperactive patients than in the controls. All right, so already, right off the bat, if you just take the glucose uptake in ADHD brains, the overall activity is just straight up lower than the control group. Absolute regional glucose metabolism rates differed significantly between the 2 groups for 30 of the 60 specific brain regions. So out of these 60 brain regions they looked at, 30 of these regions were significantly lower in glucose uptake relative to in the ADHD patients relative to the controls. And here's another example of that. And this is the absolute measurement. So they look at a particular patient, they look at a particular bin, and just ask what's the total glucose uptake. And in ADHD brains, for a lot of regions, the glucose uptake was just significantly lower. It's not entirely clear by significantly lower. So here they use a t-test. So whatever the measurement is, I don't know what the actual difference in the measurement is. They're saying that it's statistically unlikely that it was chance, that this difference was chance. When the values for the individual regions of interest were normalized, were divided by the global glucose values as described in the methods, 4 regions, primarily in the premotor and some metasensory cortex, had significantly lower metabolism in the patients than the controls. This is very important, and this is actually the smoking gun. So they took a particular patient. Let's say this is data from we'll say this is a particular patient. They added up the total glucose. They just took up all the numbers, added up all the numbers in all the bins, and then divided the activity in each bin by the total number. So now they're just looking at what's the proportion of glucose that was taken up in a particular bin for a given patient. This is by normalizing this way, they can compare the relative glucose uptake between 2 different patients. All right. So if for example the glucose uptake in this region is low, like the proportion of glucose uptake in the left posterior frontal region, the superior portion of that here, if that percentage of glucose uptake is lower compared to the percentage glucose uptake in a control patient, that says something, right? And you can do a statistical test to actually establish that's a significant difference, but that does say something. And after normalizing, this is what they found. These regions, the superior, upper, frontal, forward regions of the brain were significantly lower in terms of the proportion of glucose uptake. And this is the evidence. I mean, and it's pretty dang strong evidence given how they picked the subjects and how they really homogenize everyone. In addition, it's important to note that as the studies performed the tasks, the actual outcome, the percentage correct in the task by both the ADHD patients and the control groups were not different. So so again, another confound that doesn't exist, right? It's a non-factor in this result. All right. So absolutely important. This is a huge, huge result. Extremely important. And it's not just important because it's solid evidence that there is a significant brain difference between ADHD people and non-ADHD people, but it also establishes the specific brain regions that could be responsible for attention deficit disorder or attention deficit hyperactivity disorder. I say ADHD because that's the standard terminology today, and it also includes ADD in that. So, all right, so what's so important about these particular brain regions? Well, I don't know if they quite go into it in a lot of detail here, but these are the regions responsible. This is the somatosensory cortex. That's the top. That's the superior frontal region that's involved in planning complex motor movements, and then the frontal region is responsible for things like planning, decision making, and then the frontal lobe in general is responsible for things of that nature that require a lot of complex thought, the impulse control, emotional regulation, and so the fact that they found a significant decrease in the frontal region says it just says a lot about what ADHD is. That it's first of all, it's not a disorder of, it's not laziness. It's actually the brain not working in the regions that are responsible for important things. And it manifests as laziness. That's how a lot of people interpret it. All right, and then figure 4. These are actual images of a PET scan. These are transverse images taken from the, let's see, so short, little above the canthal-meatal line. So that's the line from the ear, the whole of the ear to the eye. So it's not quite horizontal, it's slanted a little up. We took a slice like that and showed what the brain activity looks like. The occiput is at the bottom, so that's the back of the head. It's at the bottom, that makes sense. So the front of the head is towards the top. The 2 subjects were selected as representative of hyperactive and control groups. White, red, and orange indicate areas of relatively high glucose. White, red, orange. So the left brain is clearly overall much more active than the right brain. The left brain, by the way, is the normal brain, and it is an example of a normal brain, and the right brain is an example of a hyperactive brain. And interestingly, the normal brain is just overall more active, and this demonstrates very clearly that you do have an overall decrease in glucose metabolism in an ADHD brain. All right, and the purple halo is an artifact. So pretty striking image. It doesn't quite clearly show, I'm guessing these colors are on the same scale. It doesn't quite clearly show the deficit in the ADHD brain. In fact, the ADHD brain frontal lobe looks a little more active, but this image was also taken in a region that was below the decrease in glucose activity was somewhere up here, and the image is somewhere down here. So not the best image to compare to figure 3, but still demonstrates that initial result of overall, the differences in overall brain activity. All right, so they mentioned stuff about, in the discussion about p-values, different type 1 errors, and that again, I'm a little limited in my understanding of statistics, so I won't go into that. But solid paper. Our findings support speculation that the frontal lobes may have a role in the pathophysiologic processes of hyperactivity, as do the recent results of neuropsychological tests of frontal lobe function in children with ADHD. The prefrontal areas are important biologic determinants of attention. Right? So they're saying that the frontal areas are very important for attention. I mean, if you have damage in that frontal area, you have trouble with attention. Disorders of the prefrontal, and this is what they explain next, disorders of the prefrontal regions often result in inattentiveness, distractibility, and an inability to inhibit appropriate responses. Wise's review of the literature on the premotor cortex concludes that the premotor cortex has a role in the preparation of specific voluntary movement, especially movement that depends on external cues. Defects in these regions could produce motor restlessness, particularly if the premotor areas normally inhibit other areas responsible for motor behavior in the adjacent motor cortex. For example, the premotor cortex appears to be critical in the suppression of relatively automatic responses to certain sensory stimuli. Clinically, hyperactive children commonly have difficulties because of their constant calling out in class, verbal interruptions, and in general, acting before thinking. Huge, right? This finding that they have is completely consistent with what's known about brain damages in the prefrontal regions. If you have, it's certainly known now, I guess it was known back then too, that if you have some kind of lesion in the prefrontal cortex, I don't really quite know where it is. And in fact, I have one of my closest friends was hit by a softball in the front of the head, and she actually does have a lesion, and she has ADHD symptoms. I think she might have been born with it, but it's not clear. Yeah, and that could have been the cause for some of her symptoms. But anyways, it's known, right? So what I said was anecdotal, but certainly through data, through actual scientific studies, it's known that lesions, any kind of damage to the frontal areas, you just lose inhibition, you start gambling more, you do more risky things, maybe you're more restless, more impatient, your personality changes in that way. You become more like an ADHD person. All right, and they also mentioned at some point that this study is not just consistent with what I mentioned, but even the prior studies with confounds, with the prior study with the blood flow, that study found that there was increased blood flow to the frontal regions during attention tasks, and this study is actually consistent with that. So very promising, and I found the study because I was able to find the study because of James Hallowell's book on ADHD, Driven to Distraction. Very good book. Lots of fascinating stories about his clinical experience. And there are a lot of studies in that book that I'd like to go over over the next, over the lifetime of this channel. All right, and so thank you for watching. And this is the end of the journal club, I guess. And if you want to stick around a little longer, I'll say a few things about the issue of radioactivity. So radioactivity at these levels for PET scans is not very high. And I took some notes here. Let's see. All right, so a PET scan exposes you to 25 millisieverts or MSV of radiation. That's equal to about 8 years of average background radiation exposure. So just by living 8 years, you would get a dosage of radiation like that. And over the course of a lifetime, 8 years, I would say it's not a whole lot. The way this is written, the way that I read this in this particular paper from 2010, it still sounds, it somehow, I don't know why, it just sounds kind of a little bad, but it really isn't. So, so the average American is exposed to about 3 millisieverts of radiation from natural sources over the course of a year. And it's really good news. I mean, this goes to show how safe PET scans are, because we, as we live day to day, we don't think about, oh, I'm getting exposed to radiation. It's from radiation from, background radiation from the environment is so low that it's almost impossible to tell if it has any actual significant effect on cancer. So doses this low for PET scans, since they don't have a good, you can't statistically determine the increase in causing cancer risk, or the increase in death from cancer. What they do is they take numbers that do give you that number, so really high, relatively high doses, and then literally extrapolate backwards to get a rough estimate. So 1 sievert, so 25 millisieverts times, let's see, 4 is 100. So 40 times the dose of a PET scan gives you a 5% increased risk of death from cancer. And now if we extrapolate linearly backwards down to 25 millisieverts, that gives you 0.125% increased risk of death from cancer. So incredibly low. And it's, that small percentage. And also, it wasn't clear from this paper what the increased risk of death from cancer was. I believe it means that if you get cancer, the chance that you die from cancer increases by so much percent, and that increase in percentages is caused by the PET scan. So yeah, so it's not clear if it's that or if PET scans cause an increase in deadly cancer. But whichever way you interpret it, it's a very small amount, and it's already statistically, a background level, and that 0.125% increased risk is a linear extrapolation backwards. It could be crazy nonlinear exponential decay downwards. We don't know, but it's reasonable if you're an adult and you don't have crazy high exposure to radiation. It's a pretty safe dose. Now, from the Wikipedia article on PET scans, the amount of radiation from PET scans is 12.4 millisieverts a year. So I'm guessing this number is probably more up to date, and people might have somehow managed to reduce the amount of ionizing radiation. So that's about half of what I was saying from this 2010 article. I'll put a link in the description. And average civil air crews are exposed to 3 millisieverts per year. And the whole body occupational dose limit for nuclear energy workers in the USA is 50 millisieverts a year. So that gives you an idea of, if you work in a radioactive environment, a very dangerous environment, the upper limit is 50 millisieverts a year for you to live a long and healthy, prosperous life. So the fact that PET scans are half or a quarter of that, and you're only exposed to that, once or a very small number of times, depending on whether you have cancer or whatever, goes to tell you exactly how safe it is. All right. So that's all I wanted to say, and thank you for watching.